Thank you again, and this is a very, very interesting seminar because it's all about food. In the middle of the seminar, I noticed everybody was eating and drinking. At lunch, we're eating and drinking. This afternoon, there'll be more food. So I would ask you, are you sure that all of that food from the hotel is safe? Are you sure? I'm sure it is. Now, I'd like to spend some time this morning talking about new regulations that are coming from government agencies all over the world. Now, when you have new regulatory requirements, that means that you also have to change the method, that you may have to modify the method, you may have to even develop new methods to give you new capabilities. My background is analytical chemistry, and I started my career as a professor of chemistry, and then I went to work for the United States government in Washington, D.C. as a laboratory director for NIST, the National Institute of Standards and Technology. And that's really how I began to get my interest in regulations and methodologies. So let me share some of that with you this morning. First of all, food safety is a very interesting field because everything begins really with regulatory agencies. Now, we believe that it's very important that we understand what's going on with regulatory agencies because we can't help you if we don't understand the regulations. Okay, and so we have a team called Business Development Team. I have a director that reports to me, and he spends 1 week, every week in every month in Europe. In fact, last week he was in Brussels for a whole week, talking to the people at DG Senko and the European Food Commission, the European Food Safety Authority, and so forth. And I will meet him Monday morning in Japan. And in Japan, we will visit the government of Japan to understand all their new regulations. And then following that, I will be in China with my business director, and we'll meet with the Chinese government for another week. And then that's 3 weeks out of the month. Then the last week he spends in the United States. So he constantly goes from Brussels to China to Japan to Washington, D.C. And that's the way we learn about new regulations, because these regulatory agencies come out with new regulations, which means that you have to change the method or modify the method. And in fact, as I travel around the world, the most frequently asked question to me is what are the new regulations? And how do I implement those regulations in my laboratory? So that's what I'd like to show you some examples of today. Now, if we take a look at the next level of these agencies, the people that actually create the regulations in Europe are DG Senko, and Senko is the regulatory body that passes new rules and regulations, and they are also supported by other parts of the European Commission and the European government that do risk assessment and risk analysis, toxicological studies, epidemiology studies, and so forth. But the regulations come from DG Senko. Now, they're the ones, as I said in my introductory comments, that have, since the last 10 years, 13 years now, they have set the gold standard in the world for high performance, low risk standards for food safety. And most countries use that as a reference point because, especially if in Korea, if you're going to export a food or grain or beverages into Europe, they will test against the European standard as an import. So when you export, you need to do the same test with the same standards. Now, that's sometimes very interesting. Recently, a country that's not a member of the European community was audited by DG Senko. They were audited in their laboratories to see they were audited based on the experience of the chemist, the training of the chemist, the performance of their instrumentation, thirdly, on the amount of training they had, and fourthly, on the ability to keep their methods up to the standards of 2009. This particular country got a perfect score of 0. And that means that their people were not well trained, the instruments were not maintained, and the methods were very old methods and so forth. Well, what happened is then the European Union stopped all of their food exports into all 27 member nations because the laboratories were not up to the European standards. So DG Senko stopped all imports from Europe into Europe. Well, that's the bad news, and of course that has a very large economic impact on that country because those figs and dates and nuts and grains, those are the major exports from that country. The good news is that the European Union also gave them $4.5 million to improve their training, improve their instruments, improve their methods, and so forth. And I'm happy to tell you that they are now have been reapproved by the European Union because the laboratories are now up to this high level. Okay, I say that because I have enormous respect for the standards that are used here in Korea. I just don't want Korea to ever experience this problem of not meeting the European standards. If I go over here to AQ, SIQ, in China, that stands for the Administration of Quality, Supervision, Inspection, and Quarantine. And that is their regulatory agency, and they're the ones that monitor all of the imports and a lot of exports out of China. In Japan, MHLW is the Ministry of Health, Labor, and Welfare. And some people call it the Ministry of Health. And they do a lot of interesting research in Japan. For example, Dr. Gilbert talked about hundreds of people testing for hundreds of different pesticides in foods. Well, the Japanese right now have a list that they call the positive list, and it has about 820 different MRLs for pesticides. MRLs are maximum residue limits. So they have 820 standards for pesticides and veterinary drugs in foods. And as you visit the Japanese research laboratories, you'll see that in fact their goal and their objective is to develop methods that will allow you to analyze up to 1,000 different pesticides in a single analysis. Not a single run, but in a single analysis. If you can imagine, that's a very, very difficult standard. The second thing about Japan is not only are their standards high, but for all the food that's imported into Japan, 80% is tested and inspected by the Japanese. Now, the Chinese have had a very hard time importing food into Japan because the Chinese test only a certain part of the food. The Japanese test 80% of everything. So the Japanese are actually then rejecting a lot of shipments. Now, I said that the Japanese are testing 80% of all their imports. If you go to the United States is testing 0.01% of the food imports. So now the European standard is higher here, Japanese standard is here, and the United States standard is probably somewhere in the basement of this hotel. Okay? So I'm not very proud of that. But But that's changing. And I'll show you some examples in just a minute. In the United States, those regulations are really, really developed by the Food and Drug Administration and the United States Department of Agriculture. And those two agencies cover all of the food, beverages, and agricultural products. Okay. Excuse me. So now if we look at some of the specifics of these various countries, let me start with Europe, because Europe has many things going on, as you heard in the previous presentation. The first of these is the harmonization of pesticide MRLs. Remember, that's the maximum residue limit. Now, the problem today is in Europe is that they have European standards, but each member nation in the European Union can set their own standards. So it means, in principle, that you could have 27 different standards, different norms, depending on the country. So what they're trying to do is to unify these regulations so that countries like the United States and Korea have an easier time of importing food into Europe. That's what they're trying to accomplish. Now, that means that you have to get agreement on the maximum residue limits with all these countries. And of course, that depends on technology, it depends on the economy, it depends on many factors. The next area is that of mycotoxins. And the mycotoxins today are very high and very strict standards and regulations in Europe. And I'm still talking now about the European Union. These standards are very, very high, and in fact, that's what makes it so difficult to import food like nuts and figs and dates and grains into Europe. And you heard Dr. Gilbert talk about how difficult it is to find mycotoxins in food. Usually they're not visible to the eye. Sometimes they are. The other problem is that mycotoxins are heterogeneous in the sample matrix. So your sampling has to be statistically accurate in order to get representative samples. Taking a single sample out of a batch of figs is not representative, and you may get a false positive, you may get a false negative. Okay? So now what's happening is that these standards for mycotoxins are getting so high that now you don't really have a choice except to use mass spec-based methods and new mass spec technologies to positively identify and screen mycotoxins for foods for mycotoxins. The third area I'd like to just touch on is that of nanotechnology. And nanotechnology is rapidly being developed for food in Europe and in Canada and United States. Let me just give you a few examples. Nanotechnology in foods, this usually involves nanoparticles. And the nanoparticles are usually cylindrical, right circular cylinders. And the length of these nanoparticles can be as short as 1 nanometer or as long as 100 nanometers. But either way, they're not visible to the eye. So they're invisible, and therefore you have to have special handling techniques. Now, first of all, you see some very encouraging results by using nano silver particles as a detector that's impregnated in the food and would detect the presence of a pathogen or a toxin in food. So that when the food goes to your grocery store, you then scan these particles, you scan these foods for the silver particles, and then you get a positive result, or you don't, or you get a negative result, hopefully, for pathogens and for toxins. There's another study going on that's very interesting, and that's to coat these nanoparticles with antimicrobial agents. And so if you mix this with a food, like a fruit, for example, and you blend this and mix this, then in fact the antimicrobial properties of the nanoparticles are impregnated in the food, which helps preserve the food so it doesn't spoil. For example, if you have a tomato, a red tomato, and it has a small dark spot, or we call it a bruise, something that's off color, that's usually the first sign of a presence of a pathogen impregnating a tomato. Well, the way you identify that for certainty is you impregnate a few nanoparticles into tomatoes. Well, that's good. But now the question is, you and I are going to eat the tomato, and now we just ate the nanoparticles. Okay? So that's the bigger question, is what's the fate of the nanoparticles in human beings? There are many examples of nanotechnology, but one more is a very interesting one. It's kind of funny. In the United States, there's a laboratory that finished a study to look at how you eliminate and how you kill all microbiological activity in hot dogs and processed meats. Now, processed meats are usually a mixture of many different kinds of meats and animal parts. Okay? And the example I'm going to use is hot dogs, because hot dogs are very heterogeneous. You know, it's some pork, it's some beef, and it's some other parts, and so forth. So, and then you put them into a casing, and that's what you and I call a hot dog. Well, because it's an unknown mixture of parts in a processing plant that may or may not have sterile conditions. So the question is, how do you make certain that hot dog is safe to eat before you eat it? Well, this laboratory impregnated a large number of hot dogs with these silver nanoparticles, and they determined that if you heat a hot dog at 1100 watts for 75 seconds, it will kill all the pathogens and toxins and biological activity, and it's perfectly safe to eat. Now, what happens if you put a hot dog in a microwave oven for 75 seconds? A hot dog contains a lot of water and a lot of fat. And those polar molecules have a very high absorption efficiency of microwave energy. So all the energy goes right into the fats and the waters and so forth, and pretty soon the hot dog blows up. Okay? Well, that solved the problem. It's now safe to eat, but you don't have any food left. Okay? So there's a solution to a problem that wasn't really a problem. Now, if I go to Japan for a minute, Japan, as I said, has three things that they're working on right now. One is this high level of import testing. And in order to do that much testing, they have to go from targeted compound testing to screening testing. There's no way to test that much food if you don't do screening analysis. And we're working in collaboration with quite a few Japanese laboratories to develop screening methods, and we'll talk more about that today. The second thing is that you see also the world's largest list of MRLs for food contaminants in fruits, vegetables, and grains, and also for veterinary drug residues. The third thing about the Japanese is they have a policy for food of zero tolerance. And zero tolerance means that if any of the food that you're importing into Japan tests positive for any